Thank you. Hi everyone, I'm Samaa Gobran. I'm a finishing PhD student in Dr. Nagda Shukri lab, and today I'm going to speak about HCV-HIV co-infection. I will try to be as accessible as the previous presenters. But overall, globally, about 39.9 million people are living with HIV. About 5.8% are also co-infected with HCV because of the shared route of transmission. So HIV is a retrovirus that infects CD4 T-cells. The main entry receptor is CD4 T-cells. However, there's other co-receptors such as CCR5 and CXCR4. After entry, the virus reverse transcript its genomic material and become integrated within the host cell genome. This is called the HIV reservoir, and it's a major obstacle in the eradication of HIV infection. Following virus integration, the virus will express genetic material forming new variants in the vast majority of cells that then will die. However, a small fraction of cells will persist to harbor the HIV reservoir, which are called the latently infected cells. Antiretroviral treatment can suppress HIV RNA level to undetectable level. However, whenever the patient stops the treatment, there will be a rebound viremia. This is why HIV should be treated for life. And we are focusing more on the latently infected cells. We are trying to, previous work have also shown that the reservoir is very diverse. So different antigenic specificities could contribute to this reservoir. However, we don't know yet whether HCV-specific CD4 T-cells contribute to this reservoir or not. And this is actually our research question. So we hypothesize that in HCV-HIV co-infected people, due to inflammation and immune activation, HCV-specific CD4 T-cells are susceptible to HIV infection and harbor HIV reservoir over time, even after treatment with antiretroviral treatment. To target this hypothesis, we had two objectives. Firstly, we wanted to characterize the phenotype of HCV-specific cells and whether or not they are susceptible to HIV infection in vitro. So we had a cohort of 8 HCV resolvers and 2 chronic HCV people. We tried to perform an assay that mimics the antigen presentation that happens in vivo. So it's an assay called the CFSC proliferation assay, in which we co-culture dendritic cells that are labeled with different antigens with the CFSC labeled CD4 T cells. So CFSC is a proliferation dye. So if the CD4 T cells will proliferate in response to the antigen, the dye will be diluted over time. After 5 days, we will gate on the CFSC low population in red, and these are the antigen-specific cells. So by this means, we can check for different antigenic specificities, and based on the literature, we had different controls, like Candida albicans, CMV, Staphylococcus aureus, which, based on our previous research, it's a positive control of HIV infection, and SEB, which is a positive control of proliferation. So and compared to all of these engines, we wanted to understand how HCV-specific CD4s could be permissive or not to HIV infection. But primarily, we wanted to assess the different co-receptor, HIV co-receptor expression, mainly CXCR4 and CCR5, which are a major entry receptor for HIV, and also integrin beta-7, which is a gut homing marker, the CXCR6, which is a minor HIV co-receptor, but also a liver homing marker, and CCR6, which is a Th17 markers. So we checked for all of these markers on the cell surface, and we came up with a heat map. We used an unsupervised gating technique called FlowSome to check which markers are enriched on which antigenic specificity. For example, in HCV-specific CD4 T cells, we found enrichment of cluster 4, which had high expression of CXCR6 and CCR5, which is similar to our positive control with the Staphylococcus aureus. It showed similar representation of this cluster. However, SAB, most of the cells were clustered in population 1, which has low permissiveness to HIV infection. Candida, it's known in the literature that it has high expression of CCR6, which is cluster 3. CMV showed more or less equal representation everywhere. So we could conclude from this set of experiments that HCV-specific CD4s have high expression of HIV co-receptor CCR5, CXCR6, and CCR6 compared to other engines. So as we saw that the cells have permissiveness profile, we wanted to repeat the same experiment again, but here we added HIV to the system. So we repeated the same co-culture, but now we are infecting the cells actually in vitro. And here we gated on the CFSC low, which is the antigen-specific cells, but then we looked at HIV infection within the cells by gating on the HIV P24, which is a capsid protein. So the cells that express these proteins are infected with HIV. And we also gated on the CD4 low population because after infection with HIV, cells tend to downregulate CD4 T cells. So we consider P24 positive CD4 low cells as cells infected with HIV. As you see, compared to the SAB, which had very low level of infection, most of the engine has significant infection rate with HIV. However, we can see by the significance that the Staphylococcus aureus, which is a positive control, had the highest infection rate, and this was followed by HCV. So we concluded that a fraction of the cells are susceptible to HIV infection, and they express high levels of HIV co-receptor entry markers, such as CCR5, CXCR6, and CCR6. And then we wanted to explore in real-life setting what happens in a co-infected person in terms of HIV reservoir. We had samples before and after HCV resolution, and this patient is had an outstanding history because he was infected with more than 18 years, and he had 3 episodes of HCV infection and resolution. But for the sake of this presentation, I'm only focusing on the 3rd episode. And we had 3 time points. Early acute time point, 3 months following the infection, and then follow-up time point 6 months after HCV RNA is negative, and a late follow-up time point 18 months later. Firstly, we wanted to identify the phenotype of the engine of HCV-specific cells using the AIM assay, or the activation-induced marker assay. So it's an assay that helps us to check for the phenotype of engine-presenting cells, but in a different way. We co-culture the BBMCs, the peripheral blood monocytic cells, with HCV peptides, and then overnight we check for early activation markers. This is how we gated on the cells. So we gated on the CD69 positive population, and then cells co-expressing CD4 T ligand and OX40. These are all early activation markers that are functionally defined antigen-specific cells. And then we looked into different polarization markers to check for the subsets of the cells. What I want to focus on is that, as expected, the cells goes really low after viral resolution. So we see at the early acute time point there's some cells, but then at the follow-up time point, when the virus is no more there, the frequency of the cells goes very low. There's also an interesting shift between a TFH polarization profile to a TH17 profile after resolution. So we concluded that, okay, the same patient has different frequency of HCV-specific cells and also different phenotype. If this is the case, how about the replication-competent reservoir within the cells? So we performed an assay called the viral outgrowth assay. It's the same co-culture system, however, it's extended for a long time. So the aim is to reverse the latency of this reservoir. So the red reservoir in here, if ever it's competent of replication, we wanted to culture the cell for a long time enough to have finally virions. So it's an extensive, long experiment, and we had the same engines as before, but we added a positive control, which is polyclonal activation using anti-CD3, anti-CD28. We used replicates of cells co-cultured, and we put the cells in culture for 15 days. Every 3 days, we split the media. So we start with 1 plate and finish with 16 plate. so that we can see if ever the virus is able to replicate again. And the readout is a P24 capsid protein. This means that the virus has expressed the genomic material into a real virion. So it requires a lot of intactness of the virus to be able to express itself. And we were fortunate to see in our 3 triplicates at the early acute time point some replication in here, the P24 positive cells. This means that these CD4 T-cells were able to express HIV in the HCV-specific, in the HCV stimulation wells. However, at the follow-up time points, as we saw very low frequency of the cells, we couldn't see any reactivation. It could be due to the low frequency of the cells or the complexity of the assay, or maybe there's no intact virus. We really don't know why we didn't see that. This is why we searched by other means that I'm going to explain later. But if we focus on the bright side, on the early acute time point, in the 3 triplicates and compared to other engines, the highest reactivation was among Candida albicans specific cells, followed by HCV specific cells. So compared to other engines, there is more reservoir intactness within HCV specific population. And when we do P24 ELISA, we can see that at day 12 and day 15, there is a reactivation of the virus in the HCV specific population. So this makes us hopeful. So we repeated the same experiment, but here, as we didn't see much in the follow-up time points, we need to use another assay other than the VOA. So we use a simpler one, which is PCR. However, PCR can tell you whether or not there is integration, but doesn't tell you whether or not this integrated virus is intact. But the good thing is that we have equal number of cells per well, so the frequency of the cell is not an issue. So we repeated the same experiment of co-culture, but here we added antiretroviral treatment for 5 days, and then we sorted the cells. We sorted the CFSC low, which is the antigen-specific cells, and we used the same engines, and we can see here that the Staphylococcus aureus, which is our positive control, has very high integration at all time points, and we see some of HCV-specific CD4 T-cells. There's some integration too, but if we focus mainly on HCV-specific cells, and here we have equal numbers of cells per well, early acute time point, we see some integration. However, after the follow-up time point, when there's no more HCV, we still see HIV integration. This means that even following resolution, HIV reservoir persists. And we concluded that a fraction of HCV-specific CD4 is susceptible to HIV infection. The cells express entry markers for HIV, and the cells, at least at the acute time point, have replication-competent reservoir, as we showed by the VOA. And the integrated HIV DNA persists even after resolution, which raises concerns in the co-infected population that even after resolution, HIV reservoir could persist. Future direction, as we couldn't see much intactness in the 2 other follow-up time points, we are now sequencing we are doing near full length sequencing of the antigen-specific cells to see whether or not there is a genetically intact HIV reservoir in the follow-up time points. And we wanted to assess for whether or not HIV is integrated more in specific clones within HCV-specific CD4 T-cells. This is ongoing. By this, I wanted to end by thanking my 2 labs, Shukri Lab and Ankuta Lab, who is also specialized in HIV reservoir. I would like to thank my collaborators and the funding agency, and thank you for listening.